All right, so yeah, today's webinar is to discuss the Strandings of Oceania Database, which I did some work on a few years ago when I was also seconded to SPREP. And we just want to socialize it a bit with people in the region. So what we're going to cover is just a bit of a discussion about what kind of marine mammals we get in the Pacific, what is a stranding or an incident, a look at the strandings that have happened in the region, and then we'll talk about the database itself and do a bit of a tutorial and exploration. Feel free to use the chat or ask questions at any point. If you raise your hand, I will call on you. I don't mind being interrupted, it's fine. All right, so over half of the world's 90 species of whales and dolphins have been found in the Pacific region, and at least 30 as year-round or seasonal migrants, including the large migratory baleen whales such as blue whales, fin whales, and humpback whales. They use this area as a breeding ground during the austral winter. You also have small dolphins which might be found year-round in the shallow bays or coastal areas. And some species such as spinner dolphins, short-fin pilot whales, and sperm whales are widely distributed across the region, whereas others, such as the Australian snubfin dolphin and the Indo-Pacific humpback dolphin, occur more locally. The region is also home to 5 populations of dugongs, which are also included in the Strandings database. These are in New Caledonia, Palau, Papua New Guinea, Solomon Islands, and Vanuatu, as well as in Australia. And the largest population of dugongs is in the Torres Strait. Some of the endangered species that are found here include the sei whale, blue whale, and the Oceania subpopulation of the humpback whale. So within our marine species program, there is some information about whales and dolphins and dugongs in the Pacific, including this table, which outlines whether or not a species is known to occur in a certain country or territory. But I think this kind of highlights that we don't actually know a lot about where these animals occur. So what is a marine mammal incident? An incident is a bit of a catch-all for live strandings, animals who have been beach cast, already deceased, maybe they've died at sea and have washed up, entangled animals, or ones who have been hit by a ship or anything like that. But also the term stranding is often used as a catch-all for all types of incidents. And incidents can range from one individual to many individuals, and a stranding of many individuals would be called a mass stranding. So a question we often get asked is, why do whales strand? And the basic answer is we don't really know, but there are many theories. Animals could be old or sick. So old whales can find it difficult to keep up with the rest of the pod, or to resist heavy swells or strong currents. And because of their weakness, they can strand. They can also suffer from diseases, which may leave them weak and disoriented, or with impaired echolocation, which is what they use to navigate. It can also be problems with birthing, so they may come close to shore and get stranded that way. They may be injured, which can result in stranding, or disorientation if their hearing is impaired somehow. One of the other theories is navigational errors. So There's a couple of things that could happen here. For example, when chasing prey or being chased, they could come into close area and get caught out. There's also some geographical reasons that they could get caught out, such as really gently shelving, silty or muddy beaches. There's a theory that the echolocation can't bounce off those areas very well, and so they don't realize that it's getting shallower, and then they can get caught out, especially if the tide is fast dropping, which can be in those kind of areas. And then lastly, there's social bonding, which is particularly implicated for pilot whale strandings. And the theory is that there's one individual that is having one of those previously mentioned problems. It's coming close to shore, it might get stranded, and then it calls out to its pod, and the pod follow it in. So yeah, it's very hard to know what the problem is in these situations. Most of the time we really don't know. It sometimes is a bit obvious if it's old or unwell or injured, or if there's been a birthing issue, but otherwise healthy animals stranding, it's very hard to tell. Even if you are able to do a full post-mortem examination or a necropsy, it can still be very hard to know. So a quick look at strandings in the Pacific. So this is using the data that we already have in the database. We can see it's reasonably wide-ranging, though it's worth reinforcing that this is obviously a very small amount of data that we have in there so far. It's not complete. We have a lot of great data from New Caledonia, which is really awesome. But we also have records from Samoa, Tonga, Cook Islands, French Polynesia, and Vanuatu. And the database gives us these little graphs that show the breakdown of the split, which is quite handy. So a quick note on why it's important to collect data from these events. So it's a really good source of info on species identity and diversity, and sometimes you can get a better understanding of the threats to a species or a population if you're able to find out why the animal has stranded or died. And obviously the Pacific can be very remote and inaccessible, so this is kind of an easier way to collect some of this data that you wouldn't otherwise be able to get. And as I was mentioning, the database that we're using gives us some simple data analysis. So for example, the most common species that we have recorded in the database at the moment is dugong, as well as sperm whales, short-fin pilot whales, and Cuvier's beaked whales. So a bit of history of this data set and database. So there was an original database called A Pattern of Dolphins in Oceania, or APOD, which was set up by the South Pacific Whale Research Consortium, along with this organization called WildMe, to document strandings of whales and dolphins in nations and territories of the SPREP region. And unfortunately, this database became sort of defunct as the technology evolved and it wasn't supported anymore, and there was no one really managing it for quite a while. So WildMe offered to move the data into their modern platform called Flukebook, which is a bit like Happy Whale, in that it's designed for photo identification, catalogs, and sort of AI technology in identifying fins and flukes and stuff. But it is, it's serving as a basic data record for us until a better alternative comes along. So yeah, it's just worth keeping in mind that we're not using it for its actual purpose, but it's a place to have the data and have the data viewable and explorable by people. So we've created a form that can be used by anyone throughout the SPREP region to record data. If you don't already have such a thing, it's not essential that you use it, but it might be helpful to know what kind of data is useful to gather. And this includes measurements and a guide to how to do such measurements. Here's a photo of some measurements being taken. They're always taken in a straight line rather than curving around the body. This form is available on our website to download as well. And then a bit of information about how we submit data to the database. So basically there's the website flukebook.com, and ideally you have a login, which I can create for people. It's much easier if you're logged in when you submit data. In theory, you can submit data without having an account, but there's currently a problem with being able to add those encounters to the project, but I am working with the team at Flukebook to remedy this, so hopefully we'll have some updated guidance on how to do that sooner rather than later. So you log in, you can submit your encounter using this form that they have on their website. If you're logged in with your account, I will have given you access to the Strandings of Oceania project, and you tick that. You add in location information and photos and the date, and you can even add biological samples. This is an example from New Caledonia. And once again, I'll go through all of this when we do a little run through on the actual website. And there's guidance for this on the SPREP website with just step-by-step actions to take. And alternatively, if technology is not your thing, you can send your information to SPREP and we'll upload it for you. And your main contact there will be Karen. So this is what it will look like. I'll have APOD 29. APOD is the sort of prefix for the data in the project. You have a map showing where it was, photos, and then attributes and comments. If you're only interested in viewing and using the data, to view it on Flukebook, you will need a login, which once again I can create for you. And I have also written up step-by-step instructions on how to view the data. But we've also downloaded the data into an Excel spreadsheet, which is on the SPREP website on the Pacific Environment portal. So we have the data there, the stranding form, and the protocols that I've mentioned. So far there's 76 encounters in the database. Encounters is the word that Flukebook uses, which is basically just an incident. And these range from 1991 to 2021. We're very keen to get this database completed or more representative of the region. So please do get in touch if you have any data that you want to add, or if you want a login. Otherwise, I can help get the data entered. I'm going to be with SPREP for another couple of weeks, but I will also be able to help Karen with it once I'm finished. We have some links here if you want to learn more about the database or WildMe. There's a guidance documentation on the WildMe website that talks about the wild books, which are different types of books for different animal ID. And so that's my introduction to the database and the data, and I will end the slideshow and then we'll go into the website and I'll show you around. So give me a moment. Does anyone have any questions so far? you know if you have any issues seeing that. My chat back up. Participants pane. There we go. All right, so this is the Flukebook homepage. I'm logged in, and let's first look at the project data. So there's two ways to do that. We can go my data, my projects. I've only got one project, so Strandings of Oceania. And then we get a table, and you can open up any of these to look at the details. APOD 11, it's a deceased animal. In New Caledonia, 2009. No photos for this one. Dead, the sex was unknown. Fortunately, no additional comments on this one, but it was submitted by Claire Garrigue. And then there's a biological sample at the bottom there as well. Tissue sample stored in alcohol. The other way to look at the data is to do a search. So you can do an encounter search. This takes a minute to load. I apologise. So you can search within the map or by the country, date, the species, or maybe who submitted it. But easiest for us is to look by the project. Once again, we get a table of data that's assigned to this project. And we can view the warning. There will be images of dead whales and dolphins on here. We get all the images that have been submitted. We can see the mapped results. And then there's the analysis. A lot of it is not super helpful for us, but there is the little graphs that I used in the presentation showing the species distribution and the countries. And then if you want to export data, that's there as well, just in the standard format.
So Hannah, when you create our logins and you say that we're linked to the project, we can get all of this information because our logging is linked to the project?
Yeah,
is that right?
And then there's another step where we basically create a collaboration together. So I've got collaborations with all these people, and then I can say that you can view or you can edit the data that I'm responsible for. And so once I set up anyone's accounts, I would basically send you a collaboration. Do that down here. User lookup. I would send you a request to collaborate, and then we would initiate it, and then I can say that you can view or you can edit this data.
Thank you.
And then let's have a look at submitting an encounter. And Christina has helpfully shared some data with us that we didn't already have in here, so we can use that as an example. So we have a report about a sperm whale stranding. So we're on report and encounter. The first thing to do is to add the photos. So I have handily already got all the photos. And hopefully just drag them on. Yep. So date and location, we've got the 1st of the 9th, 2020. It didn't work. It's supposed to be dashes. And the encounter location. So was this one of our location IDs? We've got all of the Pacific Ocean, or we should have all of the Pacific Ocean countries in here. So we are looking for Vanuatu. And Eton Village. Actually, you have to do that twice. And then you can either enter latitude and longitude, or you can find it on the map. Can I find, there we go, Vanuatu.
Bit further south.
Sorry?
Bit further south.
More further south?
Yeah. Yep. Further south. Next island. Bit more south.
Oh, gosh. Port Vila.
So that, okay, Port Vila. So there you go. And then you've got Eton. There you go.
Thank you. And then I can just double click and it will give me a coordinate. There will be obviously fields in here that aren't relevant for us because it's made for photo ID. That's right. I'm going to change this to Christina's details. What's your email address, Christina?
Christina at vanuatuconservation.org.
Thank you. And then if you wanted to, you can also include a photographer's name. We'll choose the Strandings of Oceania project. And then we can add in any additional comments. So we have got sperm whale, email due to size, moderate decomposition and station. And then you can include whatever measurements you want in the comments as well. I'm just going to put the length in for now. And then you could put these details in here as well if you wanted. And we have taken some samples so that we can add those in later as well. Let's leave it at that for now. Species, sperm whale. We're assuming male. Status is dead. So you can use the sighting ID for sort of alternative IDs. If you have a local ID number, you can put that in there. And comment if it has any scarring or other identifiable features. And then the life stage, which we're thinking this is an adult due to the length. These are not measurements of the animal. This is environmental measurements, which you're unlikely to have, but you may. And then if you want anyone else to find the, to get an email about the sighting, you can add that in there. And then we'll send the encounter report. Unfortunately, you have to add in the biological samples afterwards. It's a little bit awkward. That's a bit slow. Helpfully, there is a WildMe community forum, so you can post about any issues, or if you want them to add a new feature or something like that. So I've been doing that a bit to try and improve the experience for us. Apologies, it's taking a while.
And can we use the Flukebook for sightings as well as strandings?
Yep, definitely.
But that would be outside this project, would it?
It wouldn't be a part of this project, but you can use it for its actual purpose, which is, yeah, recording sightings and matching for individuals. So it's the same kind of thing as Happy Whale, if you've used that. You could, I guess, you could create, I'd have to see if there are any projects already for like sightings of, say, humpback whales in the Pacific. I'm not too familiar with that. But we could look into that, or you could talk to Karen about that. There we go, success. And Christina should have received an email as well that will give her a link to her encounter, since I put her in as the submitter.
Yep, it's just come.
Great. So now we can go and view the encounter. At the moment, it's unassigned, but I'll show you that step in a second, which we're going to give it a project ID number. There's quite a few photos, so they're all there. So me and Karen with SPREP, and then all the people in the research consortium have the ability to basically approve encounters that have been added to the project. So we can, I guess, verify that the details are correct and the species ID matches what the photos are. So then we can change this to approved. Doesn't really matter, but you know that someone's looked at it and confirmed that it seems to be the right thing. And down the bottom is where we can add the biological samples. Freaking out with all the photos. I won't put these in right now because we'll need to figure out what the sample ID is going to be. The only ones we've done so far have been for New Caledonia, and they've got their own sample IDs, but you would figure out a sample ID nomenclature, choose what the sample is. So these would be tissue samples, skin and blubber, and muscle by the looks of things too. How it's preserved. So in this case, we've got them frozen. You can record any of these other things, but most of it's not really relevant. And you just save that, and you can add as many as you want. Now I want to add a project ID number to this encounter. So I'm going to go to view my projects. I mean, you guys don't need to worry about this because that wouldn't be your job, but out of interest. scrub to the bottom, and here's our newly created encounter. And we're going to do mark new, which is basically saying it's a new individual in Flukebook. If you knew that it was an animal that had already been recognized and put in as a sighting, for example, you'd be able to match it using the photos. But in this case, we, as far as I know, don't have a good repertoire of Pacific whales, so I'm just going to mark it as new. Success ID has been added for this individual. So if we open that up again, it's now APOD 78. I think that's probably all you really need to see. I'll just show you the SPREP portal. This is where the resources are and the data you can download if you don't want to have an account and use Flukebook. You can just get the data here. There's more information on our SPREP's direct website, including the guidance and the form as well. Yeah, that's pretty much the overview and how to use it. I can make people accounts with Flukebook here on the call if anyone wants to start using Flukebook, or if you want to have a chat about sharing any data and getting it in there, that'd be cool as well. Or just let me know if you have any other questions. Otherwise, thanks for joining me.
Hey, Hannah.
So,
how many of the sort of encounters? So sometimes we see things that are posted on Facebook. Do we want to add those sort of things where we don't necessarily know very much about it? Just put it in the metadata, or
as long as there's like a date and a location, you can make a record. And then if there's any photos on Facebook, you can save them and add them. If you know what the species is, you can add that, and it's still a record. You know, it doesn't matter if you don't have all the data that you possibly could. It's still worthwhile knowing that, you know, there was this animal that stranded or died. Obviously, it's more helpful to have more information, but it doesn't mean you can't if you only have minimal information.